Okay, so we're going to start the class. We'll move on to the section which is talk about applied microbiology. So this is a pathogen control during the poultry processing. So we want to talk about this topic. Okay, the first slides. This is broiler production in the United States. And you can see the majority of the broiler production is in the southern states. So North Carolina, Georgia, Alabama, Mississippi, Arkansas, and Texas. So in other words, what this means, if you or some of you are interested about the poultry science research, then these states, the land-grant institution is a good place to go. For example, North Carolina State University, University of Georgia, Auburn University in Alabama, University of Arkansas, Texas AEM. They all had a poultry science program, and which is very good because the market is there. I used to work at Kentucky, and Kentucky had a pretty high amount, and but they tried to get rid of the poultry industry by somehow because the waste control is difficult. And our nearby states, you can see Virginia has a high amount of the poultry processing also, as well as Pennsylvania, Maryland, Delaware, and also Ohio a little bit. And surprisingly, you can see West Virginia. We are basically is a mining town, and the old time is still a little bit. But did you see we had annually 78.5 million head of the birds produced every year. That ranked about 11 among the 50 states in the United States. So that's quite amazing. And we have those small producers. So very small to small producers. So that's why we want to talk about the safety of the broiler production. Okay, what are the important foodborne pathogens? There are two. Well, obviously, any of these could be, but they basically has two, which is Salmonella and Campylobacter. These are the major two focus pathogens for the broiler industry. Okay, Salmonella, same as E. coli, gram-negative, large-shaped bacteria, and facultative. And we say it grows in the presence and the absence of oxygen, and grow better with oxygen. And the Campylobacter, this is surprisingly is a gram-positive, curved Coccobacillus. It's a fastidious bacteria. They need a special media, which is a modified chocolate media. And this is a microaerophilic pathogen, which means they only survive in the candle jar, or modified atmosphere, which is about 2 to 5% of the oxygen. If the oxygen is more than 10%, this bacteria basically is died. So the kind of thing is very ironic. Campylobacter is difficult to grow in the lab. However, Campylobacter is a major pathogen for the poultry meat. And a lot of the research has been showing the Campylobacter can exist on the skin surfaces. But in the ground poultry meat, in the poultry feeds products, the presence is almost to 0%. So it's very, very interesting. So Campylobacter we talk about, it's a curved motile rod, gram-negative. This is also gram-negative bacteria, and relatively fragile, sensitive to the environmental stress. But very importantly here is microaerophilic organism, requires 3 to 5% of the oxygen and 2 to 10% of the carbon dioxide. Now, by the way, we mentioned in our exam 1, Why this is a microallophilic? For this bacteria, the oxygen is a toxin. Bacteria survive in the presence of the oxygen needs two enzymes, which is SOD, sulfide dismutase, and also the catalase, to hydrolyze the toxication generated during the aerobic respiration. And for the Campylobacter, they have catalase, but the SOD, sulfide dismutase, is very minimal. You can look at your slides of the exam 1 when we talk about that. That's the reason it's the microallophilic. Next one we want to talk about is they widely existing in the intestinal tract from wild domestic animals, and it's very interesting. Their best surviving temperature is 42 degrees Celsius. It's not 35. This is somehow is usually the intestinal guts area temperature of birds. So that's why the Campylobacter can be survived there very well, because it gives you a very ideal environment to grow and stay there. They can survive 2 to 4 weeks under moist micro-oxygen conditions. And they also can survive 2 to 5 months at room temperature. But be careful. At room temperature does not mean at the open bench. At the open bench, they will expose to higher than 10% of the oxygen, then they will get killed. And the infectious dose is very high. It's ranged from 500 to 10,000 cells. Okay, Salmonella, we just mentioned, it's a large-shaped motile bacteria, non-endospore forming, gram-negative, can survive on the animals and in the environment, grows well at body temperature. Remember, they grow 35 up to 42 degrees Celsius. We have a terminology for that. We call this is mesophile. This temperature is very important. We will mention later on during the scattering processing. It's a very tricky temperature. Optimal temperature 35, 37 degrees Celsius. For Salmonella, slow grow at 5 degrees Celsius, which means even at refrigerated temperatures, it can grow. And our lab research has been verified, even at 10 degrees Celsius, it can grow very well. And the maximum growth between 45, 47 degrees Celsius, and slightly resistant to the acid. So they can grow at pH 4.0 and slightly at alkaline environments, which is pH 9.0. But optimal pH is definitely is a neutral pH value at 6.5 to 7.5. So that's a Salmonella. This is the top 30 serotype based on the surveillance survey in 2005. So typhimurium is the number 1, followed by epidermis. Epidermis basically is found on the egg. The Newport and Hindenburg, those top 4, you can see responsible almost 47% of the outbreaks associated with serotype in 2005. That means if we wanted to do the research for inactivation of Salmonella, then Salmonella typhimurium, Salmonella epidermis, Newport and Hindenburg should be the strain you used, because those are the target pathogen serotypes associated with outbreaks and also for the recalls. Okay, this is very important. We mentioned about earlier E. coli 0157H7 in the ground beef is 0 tolerance. We will mention again next week. But keep in mind, on the poultry carcasses, which means you buy a bag of the broiler carcasses from Walmart or from Kroger, it doesn't have to be no salmonella and no campylobacter there. So 10 years ago, the USDA FSIS, the USDA Food Safety Inspection Service, have these new performance standards. Salmonella allow up to 7.5% positive, and the campylobacter is allowed to 10.4%. So they let you have certain amount of the positive. And I can show you why this is the data they used based on the surveillance survey in the southern parts of the United States. So just want to let you know, broiler carcasses have certain level of the positive. Why? The reason is poultry usually you well cooked. When you order a poultry steaks from the restaurant, you will not say, I need either a rare poultry or medium rare. No. Because compared to the beef, they do not have a tenderization problem. It's already very tender. So that's why we usually don't undercook. But beef, we sometimes undercooked because we talk about tenderness, juiciness, and the flavor. Those we call it a palatability of the beef quality. Now, I want to mention one thing. In 2023, a new requirement comes out for zero tolerance of salmonella on a special product, which is chicken nuggets. Why? Because the chicken nuggets, who likes it? The kids. And kids is a risk group for foodborne pathogen. But that regulation comes out, makes lots of the producers really mad, which means they need to put investment for the food safety, microbial safety of the nuggets processing. For example, establish a HACCP plan. For example, following the Food Safety Modernization Act, the Process Preventive Control Regulations, which gives them a lot of the challenges by the time. So just want to let you know. But overall, broiler carcasses allow certain level of the positive. In the United States, for small producers, the processing is very verified with numerous different types. However, for the large industry, there are the standards. So we get the birds, we grow out, we live hand, we do the immobilization, we do the bleed out, we call it exquisition. Then we go through scattering process. Then we go through picking, followed by evisceration, and then go to the chilling. Now, between evisceration and the chilling, they also handing the birds back to the processing line. We call it the online reprocessing process because they're going to do the inside-outside the birds wash. And finally, they're going to do a sorting and aging and further the process the products during the packaging, and then do the commercial selling. We're not going to go over every single processing items here, but we will talk about several processing steps related to the microbial safety. So the first one is pre-scalded birds wash. Lots of the time, after exquisition, take the bleed it out, and we do a pre-scalded birds wash. So why? We want to low down the organic load entering into the scalder. And use the pre-scalded wash, we also try to decrease the bacterial amount before they go to the scalder. And we also try to reduce the feces, which is fecal samples. And we also to reduce organic load and the bacteria on the carcasses finally entering the chiller. So that's a pre-scalded birds wash the purpose. Now we're going to talk about scaldering. So first of all, scaldering is a very old processing. As early as 1st century AD, people do scaldering. Look at the sentence. The partridge is scattered with its feathers, and while with the feathers are taking off, the hair thin. So what's the idea about that? Using warm hot water to let the feather softening and easily to take it off. Easily for defeathering in the next steps. Now, scattering processing is tricky for the microbial food safety. Why? The major point of cross-contamination of chickens with Salmonella and Campylobacter, as they are liberated from the skin of the contaminated carcass and spread to the uncontaminated carcasses. So it's very important, it's a major point to prevent the cross-contamination. Because if you mix with all kinds of birds in the scatter machine, you have one is contaminated with Campylobacter or Salmonella, then they touch with each other with the skin surfaces, will cause cross-contamination from the uncontaminated to the contaminated carcasses. And that's very easy to understand. We cannot avoid that. So, what's the scattering we care about? We need an appropriate flow rate of the water. We want the water is due the direction is countercurrent, which means every time the birds is facing the clean water. And we may need to do multiple stage of pass scattering. The temperature of the scatter is very important. We'll get to that detail. The condition of the water where the birds existing is important. We want them a clean water. We don't want lots of the dust and organic matters there. And the condition of the birds at the exit is also important. We don't want to see any feasible fecal contamination on the surface. So we're going to get to the key points. The temperature control. Temperature control of the scattering is very important. So, basically, they have two types of the scattering related to the temperature. You can use a little bit higher temperature, 59 to 64 degrees Celsius, scattering 30 to 75 seconds. You also could use a little bit lower temperature, 51 to 54 degrees Celsius, but obviously using low temperature, you need a little bit longer time, so that's the time extended to 90 to 120 seconds. But there is some misconception. Number one, some people say, how about I using the temperature more than 70 degrees Celsius? Why we do that? People think I dipping a bird's carcass with feather into a hot temperature. The hot temperature could killing bacteria. However, it will cause a problem. If you are using more than 70 degrees Celsius scalding water, you will cause lipidization of chicken skin surface. So, chicken skin, what it looks like, has lots of fatty acids. Triglycerols, if you remember, this is triglycerols. That triglycerol structure, which is glycerol with fatty acids. We have stereospecific number 1, stereospecific number 2, stereospecific number 3. For the poultry, usually in the second position, this is a polyunsaturated fatty acids. So, if you are dipping birds into 70 degrees Celsius or more than 70 degrees Celsius scalding water, what happened? Fatty acids will be extracted to skin surface. This will cause a horrible problem later on during the chilling. I'll show you. So you cannot use temperature more than 70%, 70 degrees Celsius. Okay, now, lots of the small producers, they are processing limited amount of the birds. They think the temperature sometimes too high. How about if I'm using 45 degrees Celsius warm water? Because it makes you handling comfortable. What happened? I just mentioned Salmonella is a mesophile. They can grow rapidly at 45 degrees Celsius. So if you are using 45 degrees Celsius warm water to do scoldering, what happened? You are cultivating Salmonella. Then there is a joke. The scalder becomes an expensive inoculation chamber. And your cultivation bacteria, because 45 degrees Celsius, the Salmonella can grow rapidly. So that's another misconception. Another thing I also want to mention, lots of people during the scalding in the water, they also want to add antimicrobials. They think we just put the birds there, add hot water, add antimicrobials, we're killing the bacteria. That's also another misconception, because lots of the antimicrobials will be degraded into hot water, especially the oxidized agent, like chlorine. The chlorine or gas will come out. So very few antimicrobials we're using during the scalding processing. Now, importantly, we say the water flow has to be countercurrent, which means the birds come in, the birds come out, and every time they are facing is a clean water. Okay, that's a scalding. No oxidants, see that? Because high heat equals gas off, make people sick, and high organic load immediately bind and deactivate oxidant chemicals. Only strong acids can be used. Okay? So scalding temperature, scalding chemical treatments. Okay, pickers. What is important for the picker? Feeds withdrawal. Usually we do 24-hour feeds withdrawal. The feeds withdrawal time is very important. Why? If you feeds withdraw too long, let's say more than 72 hours, then the intestinal, the vet area is very soft. It's going to be broken. Then will cause cross-contamination. Now sometimes say, I'm going to do it in a very little time. How about like 6 hours? I just feed the birds and I kill them. Then all the feeds in the intestinal area. That could be a resource of cross-contamination. So pickers, very important, is for the feed withdrawal. Needs to be in an appropriate time, which is 18 to 24 hours. So, pickers. Okay, rubber pickers in the rubber fingers in pickers squeeze carcasses, making feces come out. The rubber feces on skin around and the cross-contamination from carcass to carcasses. This process may be significantly increase Salmonella prevalence and the Campylobacter numbers. And the Campylobacter usually in the ceca, you can see a huge amount, which is expressed during the pickering. Okay, the next step, evisceration. This is a major step for cross-contamination, microbial cross-contamination. You cannot avoid it. The reason is that this is the process you're using machine or manually taking all the intestinal material out of the chicken carcasses. And that is a process you basically cannot avoid it because more or less you have a bacterial contamination. But what is the key thing? You look at the picture here. What happened for these birds? the birds was not feeds for sufficient period of time, then you see the contaminated, bacteria contaminated intestinals there. That could cause contamination on the surfaces. Okay, which would cause weak intestinal, improper feeds withdrawal. So, what we do? Very important. After the evisceration, we will hanging the birds back to the surface, back to the own line. We call it OLR, equals online reprocessing process. So that's O, L, and R. And what we do is we do inside, outside the bird's wash. So we need a spray. The spray needs to be in appropriate amount of the liquid with antimicrobials, needed to be in an appropriate amount, appropriate distance, and need to be appropriate pressure. So the picture you can see from your left side works very well. That's spread evenly on the surface. So the spray time usually like 30 seconds, 20 seconds, 30 seconds. Not going to be too long. On the right side, you see some of them are not very good, and there is some of them are not even working on the top. Okay, so the pressure, the distance, and the amount is important. Now, very often what we're using is a chlorine solution. The chlorine solution, we are trying to let the pH below 6.5. We're using the chlorine concentration is usually 40 to 50 parts per million. We want the pressure can be cover up the surface and evenly distributed on the surface. Okay, we want to talk about this, very important. This figure is did by Dr. Shirley McKee. She used to be associate professor at Auburn University, Poultry Science Department. Later on, she changed the job, go to the Atlanta Chicken Council. She and her graduate students in 2015 and 2014 did the industry survey to see the chemicals used during the online reprocessing and the inside-outside the bird's wash for the chemical interventions. The number one is peroxyacetic acids. More than 40% of the producer in the United States using peroxyacetic acids for online reprocessing inside-outside the bird's wash. Followed by chlorine, followed by weak acid. For example, citric acid. For example, lactic acid. Then very few people using acidified sodium chloride, ACS. Some people using CPC, we just mentioned the centropyridine chloride. Some people using electrolyzed water, and some of them using bromine, but not too many. So lots of people, what they're using is these top three. Okay, so we want to introduce to you one by one. The first one is called peroxyacetic acids. This is a very strong antimicrobials. It's an oxidant. Peroxyacetic acid. Acronym PAA. Okay, what is this guy? This is acetic acids. This is acetic acid working with hydrogen peroxide. This is hydrogen peroxide. Generate a very special one. That's peroxyacetic acids. The key thing is this type. This carbonyl group is important. This is a highly oxidant ingredients, which causing oxidizing of bacterial cell, especially for the lipids. So where are the lipids existing for the bacterial cell? For gram-negative bacteria, phospholipids, a double layer, is that right? And also where? Lipopolysaccharides. So that's why it's very effective for bacteria to oxidize the lipids, cause lipids oxidation. Okay, so that's now the commercial products had a PAA together with hydrogen peroxide. Made a PAA about 5%, hydrogen peroxide 23%, others with water. This is a commercial product called Semi-Date 5.0. So my lab did a lot of research with this one, using for the fresh produce, and we also want to try to do on the birds processing. So that's PAA. Why PAA is the number one? Because it's allowed to be used for the organic poultry processing. Used to be chlorine is the number one, but later on chlorine is not number one anymore. The reason is, by the time when we still do the business with Russia or European countries, they're not allowed to chlorine to be used because chlorine will generate chlorine byproducts. Okay? Chlorine is a very tricky stuff. What is the tricky stuff? That's chlorine with water, which will generate. We'll care about this guy. This guy is interesting. At higher alkaline environments, this comes out. At the lower environments, at the lower pH environments, this is off gas comes out. At pH 4, this comes out at the pH 6 to 7. So it's very interesting what the curve looks like. Like this. The middle curve is this guy. At the peak value at the pH 6 to 7. So for the chlorine solution, there are some misconception. When you buy a bucket of Corax, you dilute it in the water solution. The pH is higher. How high is the pH? 10. Once that 10 pH, the dominant is this guy. This is not good. It's chlorine ions. We don't want it. We want, they have a dominant species, is HClO, hypochlorous gases. That's called free available chlorine. And HClO is a major ingredient of chlorine species will kill bacteria. The others are not. Now how are we going to achieve it then? Adjust the pH. How we adjust the pH? Pumping carbon dioxide. Then we'll generate this guy. Slow down the pH, ideally to 6.8. So there's another way to do is you adjust the pH of the chlorine solution, use citric acid to 6.8. The idea is to let the HClO dominant. So chlorine solution is very sensitive to the pH. And we don't want them to low down the pH, because low down the pH, chlorine off gas will come out. Chlorine off gas, you can feel about it. It's going to tear you. You're like crying. So I worked like 3 months at Taylor Farms in the Central Valley of California when we do the fresh produce work. We do the chlorine validation study. The whole plants, if you stay there from 8 o'clock to 3 o'clock in the afternoon, after 1 o'clock, filled with all the chlorine off gas, you're like crying there, not like you're sad because of irritation of your eyes. So that's why pH cannot be too low. If the pH too low for 3 to 4, the dominant species for chlorine solution is chlorine off gas. Chlorine have another thing is that when you're preparing a chlorine solution, the container has to be clean. If the container is not clean, the dust, the debris, we call it organic matter, they will degrade chlorine concentration dramatically. So, if you started with 50 parts per million, if the beaker or the container, which is have a dust or debris there, it will decrease to less than 0.5 ppm in 30 seconds. So when you prepare a chlorine solution, you need to make sure that container is clean, and you need to adjust the pH. What are you going to do the pH? Justification. You're not going to use citric acid to measure the mountain using pH meter. There's a pH strip we can do. And sometimes we can even add vinegar water, because that's an acid that can lower down the pH. So just want to let you know it's a very interesting topic about here, the chlorine and periacidic acids. Citric acid, hypochlorous acids, lactic acid, that's all used as a weak acids. But we don't use that too much. Why? Acid sometimes will cause a discoloration of the yellow, cause a yellow color of the chicken wings. So we don't really using lactic acid as a high amount. We control them like 2.5%. Now there is a company called Berkeley in Colorado, Denver area. They created a new product which is called citric acid and the lactic acid blender. And we gave the commercial name called Chickside, using 2.5% of the Chickside. Could have a very good antimicrobial efficacy, also will avoid partially of the discoloration of the chicken carcasses. So just want to let you know, using weak acid could kill bacteria, however, the side effects will cause discoloration. Okay. Okay, next one, chilling. What is chilling means? You put the birds, after online reprocessing, you put them onto the chilling water, which is a cold water, usually 4 degrees Celsius. And the chilling processing is usually the most effective processing to inhibit bacteria. Why? We have enough time to do the chilling. The chilling usually somehow is 1 hour, 4 hours, 6 hours, or even 24 hours. It depends the size of the birds. And because of that time, we'll let the bacteria exposed to the antimicrobials enough time. And the bacteria directly exposed to the antimicrobials in the closed container with chilling water, that's very effective. So, the research has been showing the chilling usually can decrease salmonella by 50 to 70% if operating properly. So, Chilling, we say, is the biggest hurdle because of the contact time. Dr. Shelley McKee's group also did a survey for the antimicrobials using the chilling solution. Not surprisingly, the number one is peroxyacetic acids. 35% of the people in the United States are using, followed by chlorine, about 26%. Then the weak acids used very low. Very limited amount of the people using that because of decoloration. Now, there is something I want to mention that there is several factors. You need to control the chilling water. You need to make sure the organic load is low. First of all, bleeding out. Try not to have the bleed in the chiller because that's organic matter. Second, don't put too much the feces there. So make sure the feces is cleaned on the broiler carcasses. Same thing as scattering water. The water in the chilling tank also has to be counter-current. The number of carcasses rinses is important. Number of inside of birds wash we're doing, the flow rate and direction is important, but most important is this temperature control from the scattering. Okay, I didn't remove this. And I said earlier, during the scattering, we cannot use 70% hot, or 70 degrees Celsius hot water. It will extract fatty acids to the skin surface. We call it a lipidization. If this happens, you later on put the birds into the chilling tank, and I tell you what it looks like. Your chilling tank like that. Looks terrible. This is the reason you're using more than 70 degrees Celsius of hot water to do the scattering. All the lipids from the skin surfaces of the broiler carcasses floated onto the chilling tank. It's counted as organic matters. So they will dramatically decrease the effectiveness of the antimicrobials, including peroxyacetic acids and the chlorine. And the research has been showing it will not only did not decrease the cross-contamination, it will increase it. So if you have a 40% positive salmonella birds going to the chilling tank, after you're chilling about a couple of hours, could be 1 hour, 4 hours, 6 hours, even 24 hours, then you took the birds out, you do the surveillance analysis, bacterial analysis, you find that 53% of them are positive, which means the chilling not only decreased the bacterial population, it increased the positive amount. So that tells you the temperature control in the scattering is important. Never use very hot water to do the scattering because it will cause lipidization. What the chilling water should be looks like? Should be looks like that. Very clean. Not many organic matters there. It should be look like this. Look at the clean water, not many organic matter. Okay, very unfortunately, I got these two pictures from Dr. Scott Russell from University of Georgia. He passed away 10 years ago because of the liver cancer. And he told me these two good pictures are not from United States. This one is coming from Chile, and this one is coming from El Salvador. And very ironically, in the United States, the poultry processing, you cannot find a very good example for the chilling tank. But we have to emphasize that. That's why we need to do the outreach and extension training for all those things. Okay, let's talk about in the other country. So in the United States, we're doing the chilling, is in the chilling water. In the European countries, they do air chilling. They flow with the air, the cold air, and let the hanging on the birds there, let it dry out. That's called air chilling. Sometimes the air chilling you could use in carbon dioxide, but because there is one incident that happens in some of the plants in the southern parts of the United States, so once that happens, it's never used anymore. So I just want to tell you, in the European countries, they're still using air chilling. We were very proud to say the percentage of salmonella positive broiler carcasses following chilling in the United States is the lowest in the world. It's only 5%. The percentage of Campylobacter is around 10%. This is the two surveillance results is what this standards for. Why they come out with these standards? Because of this surveillance survey. That's why I told you these slides are connected. It's very important. European Union, you get like 15% post chilling. But I want to say, European Union's results is very, very varied between different countries. In the Western Europe, in Germany, in Netherlands, you may get only 1% or 2%. But in the Poland and Turkey, kind of like Eastern Europe, you get like 70%. So you average them is about 15%. Australia is high, it's more than 35%. How about Campylobacter? Similar. In the United States, we get 10% of the Campylobacter. In post-chilling European countries, we get like 72%. Again, very, very varied distance. The standard deviation is higher, we can say. In the Australian, more than 80%. Now why? The major reason we have an in-house control system to prevent Salmonella and Campylobacter during the poultry processing. We call the terminology multiple hurdle. We have in-house